Hey guys, today I'm talking to you about protein synthesis. It is a process in which cells make proteins. There's two stages. First one is transcription, which is a transfer of genetic instructions in DNA to mRNA. The second stage is translation, is when the mRNA along with the tRNA and the ribosome work together to make proteins. We see this in the process of decoding. So decoding is a process from DNA to mRNA to tRNA and then to finding the amino acids. DNA is made up of adenine, cytosine, guanine, and thymine. In this case, adenine will bind with thymine and cytosine will bind with guanine. Now, the mRNA is made up of adenine, cytosine, guanine, and uracil. In the mRNA, the adenine will bind with uracil and cytosine will bind with guanine. The only difference between mRNA and DNA is that mRNA will have the uracil and DNA will have thymine. In this example, our DNA strand is TAC, GCA, TGG, AAT. The complementary strand will be ATG, CGT, ACC, and TTA. Now, in this example, the mRNA will take the strand out of the nucleus and into the ribosome. The mRNA strand to this will be, remember it has uracil, so A will bind to U, T to A, G to C, and so forth. CG, GC, TA, AU, CG, CG, TA, TA, and AU. This is where translation occurs. Now, the tRNA will bond next to the mRNA, and that is where transcription occurs. So it will be UA, AU, CG, GC, CG, AU, UA, GC, GC, AU, AU, and UA. You will then use a strand to find the amino acids. Amino acids are not being made during translation. They are being used as building blocks to make proteins, in which is a purpose of protein synthesis. So you will use the amino acid chart to find the amino acids in all of these codons. A codon is every 3 letters, so AUG, CGU, ACC, and UUA. In this strand, there's 4 codons. The chart will look something like this. There's also one that has a circle, but in this case, we'll be using the square one, right? So we will put this on top. And we'll use the first codon, which is AUG, which we can find here with this methionine. Then we have CGU, which we can find right over here, arginine. ACC, which is right here, threonine, and then UUA, which is going to be up here, which is leucine. Okay. Now, if there is mutations in a DNA strand, there will be 2 main types. So, missense mutations will change the amino acid specified by a codon. For instance, if this letter changed and it changed this amino acid to serine, it will be called a missense mutation. On the other hand, if a letter changes here, but the ultimate result is arginine, then it will just be a silent mutation. Okay, I hope this helped with the understanding of protein synthesis and decoding. Thank you guys for watching.